And that is, today is the first quiz. And it is available all day. But once you start, you have 15 minutes to finish it.
Yeah, I know now.
And and because you have 15 minutes that you'll take out of your time today, the class period will be only 60 minutes rather than 75. So I'll try to get done as much as I want to do in 60 minutes.
I have a quick question. Is the quiz gonna be on?
This is Yosef.
Yeah. Is the quiz gonna be on Canvas, or is it gonna be like something you just print out and then turn in like email or something like that?
Oh, you go into Canvas and you click on quizzes and click on quiz 1.
Oh, it's just fully online?
Yeah, this is entirely within Canvas.
Okay, thank you.
I also have a
They
question.
all gonna be like that?
Yes, all the quizzes will be like that. Your name, please?
Yolanda Dukes.
Okay.
And then I have a question. I'm Trinity Daniels.
Okay.
I think you might have answered this, but I can't remember. Are the quizzes multiple choice?
They're a mixture of types.
Okay.
Multiple choice, short answer, true, false, matching. So yeah, when you click into the quiz today, you'll see what's typical of all the quizzes and also the tests, of which there are 4 over the course of the semester.
Steve Stinson.
Okay.
I had trouble uploading my homework last night. And I sent you an email stating that it would not send it because I thought I had enough time to send it, but it would not allow me to send it. And so I ended up not getting it in on time.
Okay, I haven't seen your email, but I'll look for it. So what I will do is send you the number for Canvas help. And for all of you, if you have a problem with Canvas, call Canvas help. They're available 24/7. And I have found them to be very helpful when I have technical problems with that operating system. So I'll look for your email after class and send you that link.
Professor, Alex Alos. Can you hear me?
Yeah.
I just wanted to ask a quick question, which was for the worksheet that's due tomorrow for August 24th. What's the material based on? Like, what exactly are we supposed to look for that particular worksheet?
The worksheet for let's do tomorrow. Let me see.
It's methods of scientific inquiry.
It's based on what we have gone over, and yeah, it's mostly based on the materials that we've already gone over. So I think when you open it up and look at it, you'll see that there are some thought questions, there are some problem solving. It's to help you think about the material we've already gone over.
It's based on everything from last week.
Yeah.
Professor,
Uh-huh.
I was wondering, is the test that
Is this Elizabeth?
yeah, sorry. Is the test gonna be open book, or did you already?
No, all quizzes and tests are closed.
Okay. So when we click on the quiz today, is it one of those where you just can't do anything else in your screen? Like it just closes everything out?
No, there's no proctorio So
What's it mean?
you're on your honor to keep it closed book and just take the quiz. Don't look up things online.
Okay.
Don't consult. Don't get on your cell phone. So
One more question, Professor.
uh-huh.
Do you want us to take the quiz during class or after class?
Not during class. Yeah, that's why I've left it open. You have a full day from midnight till midnight tonight to do it when it's convenient for you.
Okay.
Professor,
Yes.
it's Rasha.
Russia, okay.
Yeah. Did we have one attendant for a quiz?
One attempt.
Oh, no, G.
Okay.
Professor Yoder,
Yes.
I thought it said that you can view what the test is going to be like. So what if I screwed up and it won't let me go back? I don't know what I did. It's Yolanda. I don't know. Because I thought it said you can view it when you first go on there and you're reading everything. Because I thought we were going to take the test during class today. And then it's okay, you can view it. So I'm like, okay, well, I can go on there and view what it's going to be like. And you get on there and it's the actual.
Yeah, once you click on, you have 15 minutes to answer the questions. So if you don't do well on this quiz, don't worry. The lowest quiz grade that you get will be dropped at the end of the semester. So if you miss a quiz or get a low grade on a quiz, that will be your drop.
I work on test 2.
And the tests, you can't drop.
Oh, okay.
The quests, the
Professor,
tests.
I have a question.
Alicia Carroll, okay.
Yes. On the worksheet, the very first question says list the letters in which, are we just supposed to actually list the letters, or is there like a written out answer that you want us to write to?
Any way that you can indicate that. You can circle the letters, you can write them down, just indicate the letter that it is your choice.
Okay, thank you so much.
Okay, anything else?
Yeah, this is Anthony here, Anthony Imolo. I actually have a question that's a little bit more of a side topic. During the chapter readings, one of the things it said was that animals weren't the only things that had organ systems, but that plants actually have organ systems too. And I wanted to know if you could maybe like extrapolate on that just a tiny bit.
Sure, we're going to cover that today in class. So,
Oh, cool.
Well, can I question on that one? Well,
uh-huh.
I remember reading that one as well. And I'm wondering, is that with all plants or just some? Because it had listed an example of one of the plants that you touch, and then it actually moved with your finger. You know, like when you touch it, is just some plants or all plants?
That's not the only plant that will do that, but not all plants will respond to touch. Almost
Okay,
all plants will respond to stimuli of various kinds, and touch is just one stimulus. But that's a mimosa plant that you're referring to. You've probably all done this. If you touch a twig, the leaves all fold up.
Yeah.
And you let it go and it will relax. Yeah, it's an interesting plant.
that also, you know, those little white things that you see, I don't know what they're called as a kid, because I remember doing a lot of that, and you blow them and they blow. Is that kind of like the same thing with the stimulus?
I don't know what you mean by little white things, but
you know, those little white, they look like little, when you blow them, they look like little feathers. That, you know, somebody else know what I'm talking about. I don't know how to explain it. Like little white, little ball. They like little, I don't know. You just blow them
Yeah,
and
I
then
know.
maybe you're talking about the seeds that fly around. If you blow on them,
The dandelions?
yeah, like a dandelion.
Oh, is that what they're called? Okay. So is that something
that's what they
also
call it.
considered a stimuli when you do that? Is that the same?
That's not really a stimulus.
Okay.
It's not the plant responding to a stimulus. You're providing the force to blow the seeds away.
Oh,
And the plant just sits there.
okay. By the way, I'm Yolanda.
Yeah, okay, I got you. Uh,
Okay.
Someone mentioned the textbook, so let me give you the pages in the text for the next section, which is on cell structure and function. And I'll include these pages also in the announcement for today. The textbook pages are 55 to 76, so you might want to write that down. And I will send those page numbers to you in an announcement.
Professor, I have a question.
Yes.
Do we have to take notes on these pages while we're reading them?
No. You can take notes for your own studying benefit, but you don't need to take notes from me.
Okay. And my name is Priscilla, by the way.
Priscilla, okay.
Hi, this is Sean. Was just curious. So there's a second worksheet out already, or?
Yes.
Alright.
Yeah, so you'll find that under assignments in Canvas.
I have a quick question
Professor?
about the quiz.
Oh.
Oh, sorry. Do you want to go first?
No, you can go first.
All right. Will the quizzes be auto-graded? Because I know for like some fill-in-the-blank questions, if you spell something wrong, it'll just give it to you as a wrong answer. So I was wondering,
Yeah,
like, if you'll fix that yourself or anything
I
like that.
will. I go over each question on each quiz, and anything like that, I override the grader, the speed grader.
Okay, thank you.
And what was the other question?
Yes, Professor, this is Fakria. My question is about the worksheet which is due tomorrow. I think it's tomorrow.
Mhm.
So there is a question on the last, like, what is wrong with Bob and experiment. So do we have to explain like very well written, or is just like a short paragraph, or I mean like for a few sentences, or can you please tell me like how we can write this?
You can list bullet points if you like, but look at what Bob's experiment involves. And then based on what you know about experimental design that we've already discussed in the class, just write down what you see that he either did that's wrong or he didn't do that he should have.
Okay, thank you.
Okay. I'm going to share my screen now with you. And go over the first worksheet, Introduction to Biology. And what I have here is the worksheet with answers. And I've highlighted in color the things that some of you got wrong or at least inaccurate, and I want to straighten that out. So let's just go through them one item at a time. Most of these, let me back up and go to the first point here. A few of you, most of you turned in typewritten copies, and that's my preference. A few of you sent in handwritten answers, and some of them I could decipher, and some were very difficult for me to determine what you were writing about. And so I would much prefer if you type your answers. If you do want to write them in longhand, make sure that it's clearly written so that I can read it and understand it. Okay, first, what is biology? It's a study of life. Now, some of you elaborated more on that and included viruses in the study of life. And viruses are not considered living. And we'll talk more about that and why they're not. But I just wanted to clarify that viruses are not included among living things. Then we had control variable and falsifiable. There was a lot of variation in your responses to this, but a control is a constant in an experiment. A variable is anything that can change in an experiment. And falsifiable simply means that it's capable of being shown to be untrue. You can't ever prove that something is true, but you can prove that it isn't true. Then you dealt with subdisciplines of biology. Microbiology, there are a number of different types of answers for this. It's the study of microorganisms, structure and function, and it does not include all organisms. Some of you included other things that are not microscopic, and so I made a note of that in my comment. Zoology is animals only, does not include plants. Reproduction, it's the production of progeny of anything. It's not restricted to just single-celled organisms. Multicellular organisms also reproduce. All living things reproduce. So some of you indicated that it was single-celled organisms, and I made a note of that. All the rest of these you got right. A molecule. Quite a number of you indicated that a molecule is a phospholipid. And that's not untrue. A phospholipid is a molecule. So all phospholipids are molecules, not all molecules are phospholipids. So I think you got that as an example. You read that somewhere, that a phospholipid is an example of a molecule, and then put it down as that's what a molecule is. So I took off half a point if you did that. Eukaryotes, a couple of people said that eukaryotes don't have membrane-bound organelles. That's not true. Eukaryotes do have membrane-bound organelles, and that's one of the main characteristics of a eukaryote. Talk about that later. Hypothesis versus theory. Answers were highly variable on these. I gave credit for anything that was not untrue, but what I was looking for here, the similarities are they're both testable, they're both falsifiable, and they both have a common goal of explaining a natural phenomenon. Now, the differences, a hypothesis is a suggestion or a guess, whereas a theory is established explanation based on evidence. A hypothesis is specific to one or a few observations. A theory is general, based on many tests or observations. A hypothesis is based on limited data, whereas a theory is based on many data derived from testing under diverse conditions. Then there was a question, how is theory used in lay language? And it's frequently when the media, the news media, or other sources of information talk about a theory, what they're really doing is describing a hunch or a guess or an assumption. They don't really mean theory. So I accepted a lot of variation on that. What is discovery science and what is experimental science? In a word, discovery science is observational. It's based on counting, for example. And then I think essentially all of you got the answer to fill in these boxes. So any questions about anything I've commented on, you can see my comments to your specific worksheet if you look at the worksheet in Canvas. And if you have any questions about how I graded, please send me a message. Okay. Stop sharing that. The structure of life. Anybody cannot see that? Let me know. So living things categorized as prokaryotes, eukaryotes. And this says viruses as a living thing. We're going to specifically eliminate viruses as living things and talk about just prokaryote and eukaryotic cells. So what is a cell? You had this on your worksheet. It's the smallest unit of life. Most of you got that. But I've got a note here for you to remember this inside the circle. The cell theory. You should know the three components of the cell theory. So I'm sure this is going to come up on one or more tests. So the first part of the theory is that all living things are composed of cells, one or more cells. The cell is the basic unit of life, and cells always arise from pre-existing cells, not from spontaneous generation. So make sure you know those three components of the cell theory. Then I've added a note here to emphasize that viruses are not cells. They're described as acellular, and they're not considered to be living things. Does anybody know why viruses are not considered to be living?
Is it because they need a cell to live?
Angel Arredondo, okay.
Yeah, they need like a host to live. And without a host,
They need a cell to multiply.
Yeah, you're both right.
This is Wagner.
Yes, I got you. They cannot reproduce on their own. They're not autonomous entities. So they have to be inside a living cell in order to reproduce. And we'll talk a lot more about viruses and how they reproduce, with special emphasis on the coronavirus later on. Now I want to talk about the scale of size. How do we see things from very small to very large? Starting here at the left, we have small molecules, which are measured in nanometers. A nanometer is a billionth of a meter. You can see the definitions of these measurements here in the blue box. But a billionth of a meter is very small. These are molecules. Larger molecules, such as lipids and proteins, these are known as macromolecules, are about 10 to 100 times bigger than small molecules. A virus is a collection of molecules, and that's about 100 times larger, 100 nanometers. And then we get up to into the area of organelles, mitochondria and chloroplasts, for example, which are found in cells. They're in the range of a micrometer, which is 1,000 times larger than a nanometer. Most bacteria are in the range of the same size as organelles, and about 100 times larger than that are most plant and animal cells, which have organelles, mitochondria, and chloroplasts within them. So the mitochondria and chloroplasts are about 1/100th of the size of the cells that they occupy. Now you'll see this green bar, which indicates that the items under the green bar are things you can see in an electron microscope. And I'll talk about that in a little bit. Light microscopes are not as powerful as electron microscopes, and you can use them to see anything from organelles to large cells. So a large cell, for example, a frog egg is a large cell. A chicken egg is a large cell. And an ostrich egg, it's one of the largest cells known. It's about the size of a large grapefruit. So you can see things that large with the human eye. Don't need a microscope to see things that large, or birds, or animals like humans. And then some organisms are extremely large, like a redwood tree. So this is where we can go on from here. We're going from very, very tiny things to very, very large things. And to look at even larger things, we have to go beyond the Earth. And we're going to do that. But first, I would like to show you a video.
It's natural to, when we study science, it's natural to just categorize a whole series of things as just being really, really unimaginably small. So when people say, hey, atomic scale or molecular scale or protein or a cell, you often just group that together and say, oh, those are really, 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 really small things. But what I want to do in this video is get an appreciation that even though all the things I just mentioned are really small, there's actually a huge difference in the sizes of those things. And hopefully that'll give us an appreciation for how complex something like a cell can be, how it can have all of this machinery, how it can actually be a living organism or part of a living organism. And so at this scale, this is my little rendering, my drawing of a water molecule. You have the oxygen atom in the right over here in this purplish color, and then you have 2 hydrogen molecules bonded to it. And this is going to be roughly 0.275 nanometers. And just to remind ourselves, a nanometer is a billionth of a meter. And just to get an appreciation of that, so this is 1 billionth, 1 billionth. 1 billionth of a 1/1000th of a meter. That's a nanometer. And if you want to even attempt to visualize that, that would be a millionth of a millimeter. So let me write this, 1/millionth of a millimeter. And I actually like using this one because a millimeter is about as small as I can on a reasonable basis visualize. But it's in a millionth of that. So this definitely goes well beyond at least my capabilities of visualization. So that would be the diameter or the width of a water molecule. But now let's go to the next scale up. We've talked a lot about proteins, and this is our friend, this right over here is our friend hemoglobin. And just so you get a sense of scale, the width of hemoglobin is going to be about 5 nanometers or 5 billionths of a meter. Now that seems super small, so in some ways it's okay to categorize that into your super small part of the brain, but it's good to appreciate this is much larger than a water molecule. If a water molecule were on this scale, I pre-drew it, this little thing over here, that's my attempt at drawing a water molecule at this same scale. So even when you go from something like a water molecule to a protein, you're already going dramatically up in size and dramatically up in complexity. And we've talked a lot about protein structure, how they can take on all these interesting shapes and do fairly surprising and complex things inside of biological systems. But now let's go the next scale up. And the next scale up, I'm going to go to a virus. And what I've attempted to draw here, this is a fairly well-known virus. This is HIV, and it's actually one of the larger viruses, and its diameter is roughly 120 nanometers. So if we were to draw this hemoglobin protein at the same scale as we've drawn this virus, this thing right over here would be the hemoglobin protein, and we wouldn't even be able to see the water molecule at this scale right over here. But this is still really, really small. This is 120 billionths of a meter. So this is still unimaginably small. But now let's go up to the next scale. So this creepy picture right over here, this is a T cell. This is a depiction. This is if you want to see the whole thing, that is a T cell right over here. This is a T cell. And this creepy picture, all the blue, that's the T cell. And what you see in yellow, that's the HIV virus emerging, taking advantage of this T cell. So that's why it's so creepy. It's using that cell's machinery to reproduce itself. But you immediately see on this picture how small the HIV virus is compared to the actual T cell. Each of these small little things is an HIV virus, which we already saw is a lot bigger than something like a hemoglobin protein. And so hemoglobin protein, you wouldn't even be able to, on this scale, maybe it would be a pixel, if that. And on a similar scale as this T cell, you have things like red blood cells. And this is actually a comparison, side by side. This is using an electron microscope. You see a red blood cell right over here, and you see a T cell. They're roughly on the same size, or at least the same order of magnitude size. And a red blood cell is going to be 6 to 8 micrometers wide. So this is 6 to 8 millionths of a meter. So if we were to just take 7 as the average, 7 millionths of a meter. Over here, we're talking about a millionth of a millimeter. Now we're talking about 7 millionths of a meter. And just to get an appreciation for size, we already compared the virus, the HIV virus, to this cell. We're seeing it directly as they emerge from this cell. But each of these red blood cells are going to contain roughly 280 million hemoglobin molecules. So there's going to be 200, each of these, there's going to be 280 million of these. So 280 million, that's not million hemoglobins in each one of these. So hopefully this starts to give you an appreciation for, even though we categorize cells as these unimaginably small things, they're actually far larger. They're ginormous compared to things even like large, even proteins, and especially when you think of things on the molecular or the atomic scale. And that's why cells are so interesting. They actually have a lot of complexity to them. but just to have an appreciation also for how small cells are. Even though we've just described these red blood cells and these T cells, there's these, you know, there's these kind of worlds unto themselves. They're these incredibly complex things. If I were to draw the width of a human hair on this screen right now, relative to the scale of these red blood cells, it would be about as wide as this video. So if I were to draw a human hair, it would go from there roughly to there. There's actually a lot of variance in the width of a human hair, but the width of a human hair would be just about like that. If you looked at the scale of, this picture right over here, if you looked at these scales, it would be much, 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 much bigger. And I encourage you, we can think, oh, okay, width of a human hair. Pluck a hair out, look at it, put it on a piece of paper. It's hard to even discern the width, but we're saying that width compared to these red blood cells would actually be the entire width of the screen. We've already said these red blood cells, these T cells, these are worlds unto themselves when you think of it from a viral or especially at a molecular scale. And being
Quick question. This is Trinity Daniels.
Say your name again.
Trinity Daniels.
Trinity Daniels. Okay, got it.
Will we have access to that video?
Yes, this is being recorded, so the video will be recorded along with everything else in this lecture.
Okay. All right, thanks. Gotcha.
Professor,
Uh-huh.
I don't have a question, but I have a statement. You know those little 1 million, 1 centimeter, 1 milliliter, whatever, he kept getting confused. I pray none of that go on the test.
No, I won't hold you to remembering numbers on a scale.
And it's Yolanda Dukes, by the way. I just want to make that statement. That's just, oh, God.
Okay. Can you see the screen?
I can.
Yes. Yes.
Okay. The scale of size. All right, that video was talking about everything, about the very small things, from small molecules up to the size of cells. Plant and animal cells. It was talking about T cells and red blood cells, which are somewhat smaller than most plant and animal cells. But anyway, that video was to emphasize the range of sizes from atoms, small molecules, up to cells. And the point is that cells are very complex structures composed of many, many different kinds of molecules. All right, so this slide shows different kinds of microscopes. So let's take a look at them. So first, a compound light microscope. Here's a drawing of one, a picture of one. We won't go through how it actually works, but it passes light through a specimen and then magnifies the image. So a compound light microscope will, first of all, allow you to look at living cells, and it gives you a look at the inside of cells at a gross level. So here are 4 plant cells viewed through a compound light microscope. You can see what's inside these cells. The large structures are the nuclei, one in each cell. Smaller structures are the chloroplasts, they're green. But you can't really make out much of anything else at this level of magnification with a compound light microscope. But light microscopes are very useful for many different types of observation. I want to contrast that with an electron microscope. An electron microscope can come in two versions, either a transmission scope or a scanning scope. Now, this shows you what a transmission electron microscope can do. Again, we won't go through the mechanics of how it works, except to say that an electron beam, rather than light, an electron beam is transmitted through the specimen. And it allows you to see the inside of cells, just like a light microscope does, except now you can see the fine structure. You can look at the inside of a cell at a much higher magnification. Now, this is the same magnification, just for a comparison, but a transmission electron microscope would allow you to blow this up and look at very specific points inside a cell. But now you can see a lot more. So here's the nucleus, it's a chloroplast. You can see mitochondria. You can see ribosomes, vacuole, a lot of internal structure. So remember, to see the fine structure inside of a cell, you would use a transmission electron microscope. What about scanning? A scanning electron microscope, instead of transmitting an electron beam through the specimen, it bounces the beam off the surface of the specimen. So what you see is the outside of cells. You see nothing on the inside to get a very good look at the fine structure of the outside. So these are the very same four cells, four plant cells that you saw previously, except now what you see is the fine structure of the surface of the cells. You see nothing on the inside. So again, to go back over these, a light microscope allows you to, first of all, look at living cells. Whereas the electron microscopes, you have to kill and fix the cells in order to look at them. And you can see the larger structures on the inside of cells with a light microscope. A transmission electron microscope gives you the fine structure on the inside of cells, and a scanning scope gives you the fine structure on the outside of cells. Just to show you what a scanning electron microscope can do, this is the surface of a cat's tongue. You've probably all touched a cat's tongue, you know it feels like sandpaper. It's because the cells on the surface have these little barbs. Okay, now I'd like to just take a quick mention of comparing microscopes and telescopes. So the things that are too small to see with our eyes, we use a microscope. Things that are too large, or I should say too far away to see with our eyes, we use telescopes. And there are various different types of telescope, the kind you can use in your backyard. Here's a picture of the 200-inch telescope on Mount Palomar, which is here near San Diego. There are radio telescopes, which are much more powerful. They don't use lenses, they use radio waves. There's one in Puerto Rico. And more recently, space telescopes have been developed. So they don't look out into space from Earth, they actually go out into space and travel through space while magnifying and taking pictures. And they're powered by solar panels. So this frame here in the lower right is a picture from a space telescope of just a small section of the sky. Just a very small part of the sky. And in this one image, you can see 15,000, you don't have to count them, but take my word for it. Somebody counted 15,000 galaxies are in this one little section of a picture taken by a space telescope. So what is a galaxy? Well, we live in one. We live in the Milky Way galaxy, which is shown here. Our sun is out here toward the edge of the Milky Way galaxy. But we're just one, we have one sun out of approximately 300 billion suns in this one galaxy. And for each sun, most of them have planets. And so there are more than 300 billion planets in the Milky Way galaxy. Now, the diameter of the galaxy is about 200,000 light years. So what is a light year? Light year is the distance that light travels in a year. Okay, how fast does light travel? Light speed is 186,000 miles a second. So in a minute, if you multiply this by 186,000 times 60, you get how far light travels, how many miles it travels in a minute. Multiply that number by 60 and you get how far it goes in an hour. How many hours are there in a year? It's a very large number, but if you multiply the number of hours in a year by 186,000, you get 1 light year. And that 1 light year happens to be 5.8 trillion miles. 5.8 trillion miles. The Milky Way galaxy, that's 1 light year. Milky Way galaxy is 200,000 times 5.8 trillion miles from one side to the other. So I'm just trying to give you a sense of how we've gone from atoms and molecules here on planet Earth to the monstrous universe that we live in, where one galaxy, our galaxy, is so huge. And in this image, there are 15,000 galaxies. And so this is relevant to the probability or the possibility that there might be life, not only on our planet, but elsewhere in the universe. And we're going to come back. I've got a special slide set considering that prospect that we'll talk about later. But for right now, I just wanted to give you a sense of what we see with microscopes, what we see with our eyes, and what we see with telescopes. Coming back to cells, cells can be divided into 2 general groups, prokaryotes and eukaryotes. The prokaryotes are bacteria and archaea. I'll describe these a little later. Whereas eukaryotic cells are predominantly plant and animal cells. And then we have viruses, very important in our lives, but they're not cells and they're not really alive. Prokaryotic cells are the simplest. They're very small. You saw them on the size scale, 1 to 5 micrometers. The genome of prokaryotes is always DNA. However, they do not have a nucleus. The DNA is found just scattered around in the cytoplasm in a region called the nucleoid. But it floats around there along with any other structures that are in the prokaryotic cell. And prokaryotes are characterized by the fact they do not have membrane-bound organelles. The prokaryotic cell itself is membrane-bound, but nothing within the cell is membrane-bound. Bacteria have various different shapes, but they tend to fall into one of these 3 categories. Either they're shaped like a rod, in which case they're called a bacillus, or a sphere, a ball, in which case it's called a coccus, or they're spiral shaped, as you see here. Okay, let's look at the prokaryotic cell structure. Here's a drawing of a bacterial cell. The DNA is indicated as this string. It's just a long length of DNA. And just to make a point of how efficient a cell can be, a bacterial cell is about 0.003 millimeters long. The DNA inside that cell is 1 millimeter long, which is 300 times longer than the cell itself. So the DNA in a bacterial cell is 300 times longer than the cell itself is. And that's why the DNA is coiled up in a very tight configuration. Then within the cell, there is the cytoplasm, the cell membrane, and bacteria always have cell walls, and sometimes they have flagella, little tails that can propel them. And here you see another drawing of a bacterial cell. You can see ribosomes floating around in the cytoplasm. They're not membrane bound. There's a capsule which has pili on it. There's a cell wall, a plasma membrane, and the nucleoid. Here you see what kind of a microscope took this picture. We talked about 3 types of microscope. Anybody know what type?
Scanning electron microscope.
It's an electron microscope. Which type? You remember, scanning electron microscopes show you the surface of a cell. To see the interior, you need a transmission where the electron beam goes through the cell. And so this is a transmission electron micrograph. This light area is the DNA, and the little dots here are ribosomes. You can see the cell membrane and the cell wall. Okay, eukaryotic cells, plants and animals. Eukaryotic means a true nucleus, a membrane-bound structure. The genome of eukaryotics, like prokaryotics, the genome is always DNA. The size is from 10 to 100 micrometers. They've evolved from prokaryotic cells. So here you see a picture of an animal cell. They do not have cell walls, whereas plant cells do. So plant cells usually are kind of boxy in shape. So animal cells are very pleomorphic depending on where you find them. So here you see muscle cells, skin cells, neurons, nerve cells have a very strange shape. And you saw in the picture earlier in the video, blood cells. So here's a question I want you to think about. We can talk about it next time. We're running out of time for this class. So the human body contains about 230 different types of cell. You just think about your body. You have skin cells, blood cells, nerve cells, finger cells, eye cells, all different kinds of cells. They add up to about 230 different types. Each cell contains about 23,000 genes. It's the same 23,000 in every cell. So we have 230 different types of cells. Each of them has the same 23,000 genes. Can you come up with a hypothesis to explain why different cells with the same genetic material, all the same set of genes, can look and function so differently? I want you to think about that, and we'll talk about it Wednesday. Why are we not made of just one big cell? Something else for you to think about. Why not just keep, if a living thing can be a single cell, why, as living things grow and develop, don't they just keep getting bigger and bigger cells? Humans, in contrast to a bacterium, are made up of trillions of little cells. Well, the explanation for why has to do with surface area and cell specialization. So it's because many small cells have much larger surface area than the same volume in one big cell, it facilitates transport of nutrients and hormones throughout the organism, and it facilitates communication among cells. So transport and communication is one reason that large organisms have many cells. Cell specialization is another. If you were made of just one cell, you would have one cell type. Whereas by having many, many small cells, you have 230 cell types. Okay, animal cell structure. We'll talk about many of these structures later in the course, so this is just an overview. All cells have nuclei, endoplasmic reticulum, ribosomes, mitochondria, peroxisomes, plasma membrane, Golgi. What animal cells have that plant cells don't have are flagella, a centriole, and a lysosome. So this is a drawing of an animal cell. Here's a transmission electron micrograph of an animal cell. You can see various subcellular structures, cell membrane, lysosomes, endoplasmic reticulum, little dots are ribosomes, and mitochondria. In contrast to plant cells have many of the same things. A cell wall is something that plant cells have that animal cells don't have. Chloroplasts, only plant cells have chloroplasts. And plant cells tend to have a large central vacuole that animal cells don't have. So think about that in view of the fact that the content of this vacuole is almost entirely water. So why do animals not have a big bag of water inside, whereas plant cells do? And I have indicated here at the bottom hint for the explanation, animals can move around and search for water. Plants can't, they're stuck in place. So they have to wait for water to come to them. And when they do have access to water, they store it up in a reservoir so that when the water outside goes away, they still have some inside. Okay, quickly, we're out of time, but I'll quickly run over what's left here. Viruses are not cells because as you pointed out earlier, they can't reproduce autonomously. But they're very important to us because they invade and reproduce inside other cells. And when they do that, they can cause disease like the coronavirus does. Viruses are very small compared to the cell that they invade. So here you see HIV virus inside a human cell. Examples of viruses, they can be rods like tobacco mosaic virus. They can have a complex structure like a spaceship, like T4 bacteriophage, or they can be spherical, complicated structures like HIV virus. This reiterates the same thing. Now, one thing I want to emphasize here is in contrast to cells, which always have DNA as their genetic material, a virus can have either DNA or RNA. So for example, tobacco mosaic virus has RNA, T4 has DNA, HIV has RNA. Coronavirus has RNA, and as you probably know from the news, the vaccine against coronavirus is based on RNA, not DNA. And we'll talk a lot more about that later. Here are pictures of coronavirus, a scanning electron micrograph with artificial color. Here is the little yellow dots are coronavirus attacking a cell. And here is an elaboration of the structure. Again, I emphasize, I've circled here that the genome is RNA, and this spike protein is critical for the ability of the virus to infect a human cell. And I've got a whole slide set on coronavirus that we'll talk about later. So now we have some example problems that I'm out of time, so I'm not going to go over them. I'd like you to check the video or the slide set. These slides are all in your Canvas container. So you can look at either the slide set or the video and see if you can answer these questions, and we'll talk about them on Wednesday. So here's one of the questions. You're looking at a cell under a compound light microscope. Here you're looking through a microscope at something that's rod shaped. And here you're viewing a structure that's less than 0.2 micrometers. And then answer the question, what type of microscope or what is the entity that you're looking at? And this is just a structure, a picture, a preview of when we'll talk about cystic fibrosis and the pancreas and the various different kinds of cells within a pancreas. So I'm out of time. You have a quiz to take today. Some of you have taken the quiz already.
Excuse me.
Yeah.
My name is Wasan Rafo. So we're going to see that PowerPoint on the announcement?
The what?
On the module, the PowerPoint that you are showing now, we're going to see it on the module.
Yeah, if you click on Home in Canvas, you'll see module.
Files.
Module 2. It should be in module 2. So you'll see the slide set, and then as soon as the video recording of this lecture, as soon as it's processed, which will be in about an hour, I'll send an announcement with the link to the video.
Okay, thank you.
Okay. As I said, I've gone over time. Try not to do that, but I'll stop sharing my screen. And for those of you who haven't taken the quiz, please do. And I will see you again on Wednesday.
All right, thank you, Professor.
All right, thank you. Have a good day.
Professor, thank you. Bye-bye.
Professor,
I got a question to anybody in the
I'm
class.
trying to get my question in, Mr. Yolanda. So Wednesdays, to get on the Zoom would not be the same as the one for today, right?
That's right. You have the
Okay.
link for the Wednesday class.
It's a different one. Okay, I want to make sure real quick. Okay.
Uh-huh. Yeah, and Steve, you had a question?
Yeah, my question was, anybody got an idea of how I would be able to download the worksheet to get it sent through so I don't have to jump through hurdles to email it?
Yeah.
Copy it to your device.
You can, I've given you in the syllabus, you can find Canvas help, the number for Canvas help, or I can send it to you. And I'd recommend that you or anyone else who's having trouble with any part of Canvas, call Canvas help. They are available 24/7, and I have found them useful. So I'd recommend that, and they will tell you how to submit it through Canvas so you don't have to use email. Email is very clumsy for me, for me to take your assignments.
Clumsy for me too.
Yeah, so neither of us like it. Yeah, so Canvas help can do that for you.
Thanks.
All right, I'm going to end the session, and as I said, I'll see you Wednesday.
Bye, everybody.
Bye.
Bye.